When it comes to basic immunology, we have innate and adaptive immunity. Three lines of defense protect us from foreign substances: bacteria, viruses, fungi, protozoa, and worms, termed antigens. The innate immune system, comprised of macrophages, neutrophils, dendritic cells, and natural killer cells, NK, that can travel through the blood and lymph systems and burrow into tissue to meet the antigen, where they can engulf and destroy bacteria and cellular debris. The adaptive immune system, comprised of T cells and B cells that make and secrete protein antibodies that recognize specific foreign molecules, handles viruses which do their damage inside of host cells. Cells associated with innate immunity: polymorphonuclear leukocytes, PMNs, polymorphs, neutrophils, very aggressive, short-lived cells with characteristic multi-lobe nucleus. Epithelial surfaces. Epithelia impose a physical barrier between our body and the external milieu. Epithelia comprise of the skin and linings of the body tubular structures, gastrointestinal, respiratory, and urogenital tracts. The skin. The outer layers of the epidermis consists of the stratum corneum, which itself consists of dead corneocytes, full of keratin, separated by lipid layers, Landmann units. This creates a waterproof, generally impermeable layer. The keratinocytes, if damaged, produce TNF and IL-8, leading to inflammation. Mucosal surfaces. Mechanical protection is via a layer of mucus in many instances. In the airway, the mucociliary escalator moves mucus and trapped particles to the throat to swallow. In the stomach, the mucus protects the gastric mucosa from stomach acid and enzymes. In the uterine cervix, protects the uterus from bacterial invasion. Breach by old-style IUD threads could sometimes lead to PID. On the eye's corneal surface, protects the corneal epithelium. T cells. What happens if a virus makes it into a cell? Antibodies cannot bind to them anymore to prevent its entry. Something must be able to recognize a virally infected cells and eliminate it. There are many T cells in a person and many different kinds, including T helper cells, TH, cytotoxic lymphocytes, CTL, and T regulatory cells, TREGs. T cells also recognize antigens, but unlike B cells, these antigens can only be proteins fragments. The T cell receptor recognizes and binds simultaneously to the foreign protein fragment and to the self-protein, MHC, on the surface of the APC. Pattern recognition molecules. Immune cells sense damage to foreign substances by pattern recognition receptors, PRRS, which bind to molecular pathogens common to pathogens, termed pathogen-associated molecular patterns, PAMPs. Two main classes of PRRs. 1. Cytoplasmic, for example, NOD-like receptors, NOD1, and 2. 2. Membrane-bound, for example, C-type lectins and toll-like receptors. Natural killer cells kill virally infected cells or tumors that have reduced MHC class I expression. Inflammatory mediators, the chemokines. group of peptides responsible for their ability to stimulate the migration of cells, most notably white blood cells, leukocytes. Chemotaxis. Upregulated by several cytokines involved in infection and inflammation, importantly G interferon and TNF-A. Chemokines have been classified into four main subfamilies: CXC, CC, CX3C, and C. These are produced by a number of cell types and affect mostly leukocytes, especially lymphocytes, via chemokine receptors. Chemokine cell migration through venules from blood into tissue and vice versa, and in the induction of cell movement in response to a chemical chemokine gradient by a process known as chemotaxis. Complement. The complement system is a part of the immune system that enhances the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism, promotes inflammation, and attacks membrane. Part of the innate immune system. Made up of a number of small proteins found in the blood, in general synthesized by the liver, and normally circulating as inactive precursors, proproteins. When stimulated proteases in the system cleave specific proteins to release cytokines and initiate an amplifying cascade of further cleavages. An effector mechanism designed to kill target cells and regulate inflammatory responses by enhancing phagocytosis and WBC recruitment. Consists of 30 soluble proteins. Many components are secreted as inactive precursors, zymogens, and are activated by proteolysis. Complement activation proceeds by three different pathways. 1. The classical pathway, activated by antibody attached to antigen. 2. The alternative pathway, activated by cell surfaces unable to inhibit complement. 3. The lectin pathway, activated by mannose-binding lectin, MBL. Key points about complement. Discovered in 1890 by Jules Bordet as a heat-label component of plasma, which augments the opsonization and killing of bacteria by antibodies. Opsonization is coating of pathogen by antibodies or complement proteins, so it is readily destroyed by phagocytic cells. Complement also influences adaptive immunity, T and B cells. The complement system recognizes features of microbes and marks them for destruction for phagocytes by C3b. Key points. The human body is susceptible to infection by pathogens which reside and replicate in different anatomical locations. Epithelial surfaces provide a barrier to pathogen entry.